Hey everybody, happy Wednesday. This is Chrissy Matthews. I am the 3-day program manager here at Susan G. Komen, and I'm actually coming to you live from Komen headquarters down in Dallas. So I want to thank you guys all for taking time out of your Wednesday and joining us. The purpose for today is we want to show you guys the impact that you're making with all of your participation and your fundraising that you're doing with the 3-day and other Komen events. So we are going to, for the first time ever, host a Susan G. Komen mission webinar for you. I want to point out a couple things before we dive in. If you look to the right of your screen, you'll have a place where you can type in questions. So as we go through the presentation, you have questions, please go ahead and type them in. We'll have time to address a couple of them at the end of the session, and then we'll follow up via email for any questions that we don't get to today during the webinar. And then also at the end of the presentation, we will be sending out a post-webinar survey where we would love your feedback. This is something that we're looking to do moving forward, so any insights you have would be greatly appreciated. I am joined here today by my fabulous colleague, Victoria Velochko. Victoria is our VP of Research and Community Health Program Operations, and I would like her to take the stage.
Well, thank you very much, Chrissy, and thank you everyone for joining us today on the call. Our purpose today is to introduce you to Komen's research program, and specifically one of the projects that we're funding through the support and the fundraising that all of you are making happen. As you know, supporting research discovery has always been at the heart of Komen's vision of a world without breast cancer. And as the largest private funder of breast cancer research, Komen has led the way in research investment, starting with our very first grant for $28,000 back in 1982. As you all know, research is a lot more expensive than that. Since then, we have invested nearly $890 million in more than 2,500 research grants and programs, which include about 450 clinical trials. So with such a bold goal as a world without breast cancer and millions of dollars at stake, Komen has a great responsibility to ensure that the funds our communities raise are invested in the most promising, the most groundbreaking, and game-changing research that will have the greatest benefits as soon as possible. So for us, that means we want to support research projects that have the greatest potential to reduce incidence and mortality within the next decade. This is a very, very aggressive goal. Like many organizations that fund research, we utilize a process called peer review, bringing together the world's leaders in breast cancer research to evaluate our grant applications. But what is unique about Komen's research program is the inclusion of the ultimate beneficiary of research discovery into the selection process, and that means including our breast cancer patients and our survivors. Now this starts with our strategic planning process. At the very beginning, a survivor works alongside our scientific advisory board who guides our investment strategy. This extends to the peer review process, where a survivor participates in the evaluation process of every grant application we received. In this past year, that was nearly 1,000 grant applications, and that meant that a survivor reviewed every single one of those. And finally, when we award a research grant, we require that a survivor serves as a member of every research team. It's critical to us that we always keep in mind those who will ultimately benefit from the work we support through our research program, because you know all the statistics. This year, more than 40,000 women and men will lose their lives to breast cancer. So we need continued investment in research, because although our successes have been great, these numbers are still unacceptable. And today, we say that we're within the generation of the cures. We're standing on the shoulders of the groundbreaking discoveries made in our early days that have unlocked many of the biological mysteries about breast cancer. And it's through your continued support that we can support our emerging leaders in research today so that they can find the cures of tomorrow. So I'm very pleased to introduce our guest speaker today, Dr. Justin Balko, who has an array of very impressive titles in his early career. He's currently an assistant professor of medicine and cancer biology, and he leads the molecular oncology team at the Center for Cancer Therapeutics at Vanderbilt University in the great city of Nashville, Tennessee. So Dr. Balko, are you ready to take it forward?
Hey, I am. So I wanted to tell you guys a little bit about, why I chose to do breast cancer research, just a minute or two, just so you get a little bit of a background on me. So actually, it took me quite a while, but once I was in the field of cancer research, I remembered that I actually had the goal of being a cancer researcher at a very young age. I was always a little bit of a problem solver, so I was excited by the challenge of something that other people said you couldn't do, or if my sister lost something in the yard, I thought that I could find it, and so I always considered myself somewhat of a problem solver. And obviously cancer is a very big problem. This it was also very enticing to me because there's a lot of experts in the field. There's a lot of, you see it all over the news as a kid, cancer, what's happening in cancer research, what's happening, in new technologies. And I sort of felt like the kid and the sword and the stone, right? So many people are trying to draw, this sword out to cure cancer in some respect. And so I was probably a little bit of a cocky kid, and I thought that, well, I'm going to march right up to that stone and pull it out. And so I actually left this for a little while. As a teenager, I got interested in other things. I wanted to be a rock star. I wanted to, have my own band or maybe make a lot of money or whatever. But ultimately, I think my childhood goals kind of came back to me whenever I was an adult. And I kind of got led down this path of first starting to do research and then starting to focus on oncology, and then furthermore focusing in breast cancer research. And well, why after all of those things, why after I chose cancer research did I choose breast cancer research? Well, there's a lot of opportunity, and I think, this is really in part due, to the fundraising activities of Komen and all the participants that help and get together and able to organize. Because as you may hear on the news, the funding environment for research is at a low for a number of different reasons. There's a lot more people doing research, there's a lot more areas of research, but also, just in general, the budgeting and funding is low. And so because of private organizations and because of, participants and donors such as yourself, these are what draw people into the field because there's opportunity there. There was also a little bit of opportunity in the fact that breast cancer was sort of a half-solved field. There was this notion of breast cancer kind of being on the run, and I show this in a plot on the right side of the slide, kind of showing incidence or ratio of deaths, mortality, to the number of patients that get breast cancer each year, and you can see that it's declining substantially, and this is very very dependent on the very groundbreaking research of others who have come before and are currently in the field. Unfortunately, I'm still a little bit cocky, so I still really think that we can make a serious contribution to this and continue this downward trend even further. What did all this lead to? Well, support that I got from Susan G. Komen, both as what's called a postdoctoral fellowship and during my time as an assistant professor here at Vanderbilt, was the seeding of an independent research program. So when funds are administered to those of us in the research community, they don't just go to us, and they don't just go to pipettes and test tubes and things like that. They actually go to the training of the next sort of generation of scientists, the people that are going to come after us. And that's why I have independent start here, because if I had my own lab but I didn't have other people that I was training and mentoring that were benefiting from these sorts of activities and funds, then really the research would stop right there. But so it's not just an impact to one person; it's an impact to sort of many people across the field, and including those of us that were training in our lab. Now I want to talk a little bit just sort of on a 30,000-foot view of what our laboratory does. We try to focus on triple-negative breast cancer of all breast cancer subtypes. And so triple-negative breast cancer defines those breast cancers that don't have what we call hormone receptors, which are the estrogen receptor or progesterone receptor. And they're also those breast cancers without substantial expression of what's called HER2, which is the target of Herceptin, trastuzumab, as well as a number of different drugs that have come down the pipeline in the last 10 to 15 years. So really you can see from this little Venn diagram on the right side of the slide that triple-negative breast cancer is a small proportion, but it's also really the worst outcome of all of the clinically recognized subtypes. So it tends to impact younger patients. It tends to have a very aggressive course, and there's really no targeted agents approved, and unfortunately because of that, it's currently only treated with chemotherapy. Our laboratory really focuses on two very basic but important questions. So number one, how do we identify patients that will benefit from treatments? And how do we identify them using a precision medicine strategy? So what this means is if you have all the patients that maybe are triple negative, and there's a new therapy coming down the road, and we know that therapy is effective in maybe 20 to 30% of the patients with triple negative breast cancer, we want to know, how do we pick those patients out up front? There's two things that really guide this. That's the cost of success and the cost of failure. And so if you think about not only the cost of success goes way down when you're able to pick the right patients who will benefit for a treatment, but the cost of failure also goes down when we use this strategy, which means that the patients that are put on a therapy that they ultimately don't benefit from can be stratified or changed over, triaged to a different therapy that maybe they're more likely to benefit from. So that's sort of the first question, is trying to identify which patients up front will respond to a certain therapy. But number two, and I think, to me, that both of these are important questions, but this is the one that really sort of excites me, especially in the context of the types of therapy that I'll be talking about in a few minutes. This question is, how do we make more patients respond? So when we have a really great therapy that comes down the pipeline, it's being tested in clinical trials, and maybe we only have 10% of patients that are responding to a certain drug, but those responses that they're having are really amazing responses. Well, that's very exciting, so we want to figure out how to make more patients have those kind of responses. And the type of drugs that we're studying right now are something called immunotherapy. And immunotherapy is something you might hear about quite a bit in the news as of late because of its successes in a variety of different cancer types, and we're really exploring this, whether these drugs will be effective in breast cancer. And so immunotherapies have had durable responses, and what I mean by durable responses are these are not your average short-lived 6-month to a year extension of life in patients. These are cases where patients are with metastatic disease are being almost completely cured or have substantially improved survival. And I'll show a couple of slides on this in a few moments. And so we really try to do this by combining drugs. We try to find ways to combine these drugs in order to make more patients respond. As I sort of hinted to, we have become very excited in this concept of immunotherapy. So in the 21st century, we've really been focused on molecularly targeted therapies. You may hear those buzzwords. These are in contrast to chemotherapy. So with chemotherapy, we know that these are kind of like poisons. They're toxic poisons that selectively kill cancer cells, but there's a very sort of fine window there. But in recent years, we've been focusing on molecularly targeted therapies, and these are things like endocrine therapies or Herceptin that have a really specific target. The problem is these drugs are costly and work only in a fraction of patients, and many of those patients ultimately develop resistance. So these new immunotherapy drugs are really exciting. They've had several approvals by the FDA. They're not yet approved in breast cancer, but they produce longer and more durable remissions in some patients. And so part of the reason why these remissions are much longer is that they help to engage the body's own immune system against the tumor, and they frequently employ something called T-cells. So I'm going to tell you a little bit about T-cells. So T-cells, or cytotoxic T-cells, are what we like to think of in the lab as a messenger of death for tumor cells. They're part of our immune system. What's really cool is that these cells scour your entire body constantly. So they're sort of like vigilantes that are always searching for virally or bacterially infected cells or even cancer cells. And so once a T-cell finds, for instance, a tumor cell, shown in this image right here, it attaches to that tumor cell. You'll see a little video here where in green is a T-cell that's found a tumor cell. You can see that it's sort of using parts of the cell to kind of feel out that tumor cell and determine that it's not normal and it's not supposed to be there. And once it identifies that tumor cell, then it injects poison into the tumor cell that will kill the tumor cell, and then the T cell will leave and it'll go on and continue to kill other tumor cells. And so once you get a really strong activated T cell response, you can actually eliminate cancers in many patients. And this has been studied for quite some time, but only recently with newer drugs has this been possible to employ these sorts of therapies in patients. One of the things that we think about in breast cancer are looking at these T cells in the tumors of patients. When we look at a tumor, and so this is sort of up in your upper right-hand corner of the screen, this is a cross-section of a tumor taken at the cellular level with a microscope. And each time you see a little brown dot, this is a T cell. So we've done a stain, an immunostain it's called, for T cells. And you can see that it's really interesting here when we've outlined the tumor with a dotted line, that most of the T cells are outside of the tumor, and there's very few T cells that are actually what we call infiltrating in the tumor. So this suggests that there's a whole bunch of T cells that are just kind of waiting on the outside. They're kind of like, you know, the army ready to charge, but there's very few soldiers that are actually in the field. And so when we use these new immunotherapies, and these are some more pictures at the bottom of the same sorts of tumors, where beforehand we have very few T cells, the pre-treatment on the left, we have very few T cells in that tumor. But when we use immunotherapy against these tumors, you start to see more and more brown spots invading into that tumor region. And finally, after a week or two later, what you end up seeing is that the tumor shrinks down to a very small size, and pretty much everything that's left within that tumor region is all T cells, which means there's no tumor cells. It's not even really a tumor anymore. But what's neat about this is that this activity precedes tumor regression, which means that if we've given these patients only 1 or 2 treatments, we might not see any change in their tumor size, but we can actually look at these infiltrating T cells within their tumor to determine whether or not they're going to have a substantial response to the therapy. Or if we don't see any T cells coming into the tumor, we know that the patient is unlikely to respond to that therapy. We could switch them to something else. Right now, what we're trying to do are take some of the combinations of therapies that we're used to working with in the lab and combining them with immunotherapies in a clinical trial. So we just published a paper several months ago with actually another Komen recipient, Shireen Loy in Australia. And we developed a combination of a drug that's actually used in melanoma along with a new immunotherapy, and we found that this is very effective in getting these T cells into the tumor. And so we're currently trying to bring this into a clinical trial right here at Vanderbilt to determine whether this combination of therapies is going to be very effective in breast cancer patients with triple-negative breast cancer. We're also trying to further sort of develop using these T cells infiltrating the tumor as a biomarker to determine which patients will benefit from these sorts of therapies. And then finally, what we'd really like to do, which sort of feeds back into our clinical trials, is develop some imaging markers that can allow us to not draw biopsies from patients, but use PET scanning, so something we always use for imaging in cancer patients and many other diseases, to try to monitor what the T cells are doing in the tumor. And so in the right here, just to give a minute or two of explanation, this is actually a mouse with a tumor, and it has, and we've done PET imaging for these T cells. So you can actually see them with the imaging without actually, injecting the mouse or doing anything invasive like drawing a biopsy. And you can see around that circumscribed tumor region that we have a lot of signals. So that's a lot of T cells that are just like that image that I showed you in the last slide, where they're kind of the army poised to march against the tumor. And after we've administered this combination of immunotherapy that we're trying to start our clinical trial in, we actually see a substantial increase of all of those T cells just moving right into the tumor. In this particular model, this tumor will completely regress, and that mouse will never take on another tumor. We can re-inject it with new tumor cells, and that mouse will be completely immune to that type of tumor being re-injected into that mouse. So it has a very long-lived sort of response. And so we want to try to develop these imaging markers to incorporate into our clinical trial. What I really want to say right now is how important it's very exciting, and I love talking about the research that we're doing. We wouldn't have that opportunity if it weren't for fundraising organizations like Komen and the people that participate in those kind of fundraising organizations like yourself. I can't stress enough that these provide very important opportunities that are so scarce in times of low federal funding. Not only because they allow us the research dollars to do the specific funded project, but they also give people like me, right, who are younger investigators, sort of this independent, private investment. So when these funds are available for me to start up a project, not only does it help that specific project, but it gives the institution that I'm employed at the idea that I'm worth investing in. When private foundations or even public foundations make that investment in me, as an investigator, it allows me to hire people, it allows the institution to have more confidence in me, and it just creates sort of this synergistic seeding of a research program that's ultimately going to benefit patients. So I can't stress enough that the funds that we receive from Komen and from these sorts of fundraising organization events really make or break the development of these early phase breast cancer researchers like myself. And I really like working with Komen because we have great close and very direct contact with our scientific grants personnel. I trained in a lab that worked very closely with another lab of whom one of the postdocs went on to end up working for Komen and being the scientific grants personnel on my own project. So I already know Jamie very well, and so we communicate a lot, and we're friends, and so it's great to have those kind of close relationships with the people that are managing your grants. My acknowledgments, I always acknowledge all the funding sources that we always get, but most importantly for this, especially the focus of this talk, I cannot provide enough acknowledgments to you, the people that participate and take time out of your busy schedules. We have a lifestyle here as researchers where we really don't do much fun stuff because science and making discoveries and working is the fun stuff. And I think that you guys probably understand this, that sometimes you need to roll up your sleeves and get a job done. And I am extremely appreciative that there's other people in other fields with other commitments that are able to take time out of their day and do similar things. And I just hope that from this presentation you got an opportunity to really see how that impacts the lives of researchers and the careers of researchers around you. So thank you very much to you and your friends and family for your continued support, and to Susan G. Komen. Thank you.
Dr. Balko, thank you so much. That is an awesome presentation and a good dive into what you're focusing on, so I really appreciate it. We have just a few minutes left, so there are a couple questions I wanted to toss your way.
Sure.
One of them is immunotherapy being tested in other states?
In other, like, physical states, like like USA?
I would assume so, yeah.
Okay, yeah, so we are not, for sure, we are not the only people doing immunotherapy studies in breast cancer. So immunotherapies, in the ones that I'm really referring to, a certain class of them are already approved in melanoma, lung cancer, and some like Hodgkin's lymphoma, and renal cell cancer as well. So the study that we're doing is actually, so in breast cancer, in triple-negative breast cancer, early trials, very small trials have been done, and these drugs were effective in about 10 to 30% of triple-negative breast cancer patients, which is great. That's phenomenal because it's non-chemotherapy and it's working in triple-negative breast cancer. But our trial combines those therapies, and we think that we can make more patients respond. And so at least with the early phase trials, that particular combination will presumably only be done at our institution, although we're looking at opening that up as a multi-center trial, which means we would collaborate with other institutions across the U.S. and have it done in multiple states. I think that gets to your question.
Thank you. And then there's one related question. Is immunotherapy the same as a vaccine, like what we would use for measles?
So this is like a really big nomenclature thing that's a little bit confusing. On some levels, it is similar. the immunotherapies that I'm talking about are actually monoclonal antibodies that are just, they're like Herceptin, except they recognize something else. They recognize something on T cells that keeps them from becoming activated to the tumor. Now, vaccines, as you know, are usually viruses, like attenuated viruses, where you're basically causing the body to have a major immune system to prepare itself for the real virus should you ever become in contact with it. And that's what most people think of as vaccines. But in cancer vaccines, it's a little bit different. So it's not quite the same as measles. Usually cancer vaccines are sort of a similar idea, but you can actually give cancer vaccines to patients that already have cancer. It's really just about helping the immune system activate against the tumor. So in that respect, vaccines are an immunotherapy for sure, but they're not quite the same as a virus or a bacterial vaccine.
Thank you. And then one more real quick, and actually I'm going to direct this one to Victoria. We were asked if we would repeat the amount donated to research and how many clinical trials are currently being conducted.
So since the beginning of our research program, Komen has invested nearly $890 million. That's more than 2,500 research grants and programs, and that includes about 450 clinical trials. It's important to note that it's not only grants that we support for projects like Dr. Balko's, but our research investment also goes to support things like infrastructure, serving in partnerships with other organizations that share our mission, building things like our Komen Tissue Bank, which is the only repository of normal breast cancer tissue in the world. We invest in things like this. For example, for Komen Tissue Bank, if we understand how normal breast tissue works, it gives us some insight as to why things go wrong and why breast cancer starts. So it's a variety of investments that you'll see that we've made with, which is going to be coming up on $1 billion in a couple of years. If you want to know more about some of the investments that we're making and the specific work that we're doing, if you look at the Komen website under what we do and click on research programs, there is a wealth of information there. There is interviews with a lot of our scientists, just like Dr. Balko, who will tell you more about the work they're doing, but there's also fast facts, little bullet points that you can put on your fundraising pages or on your Facebook to illustrate the impact of the research that we're supporting, not only through grants, but also through some of our programs.
Awesome. Thank you. Okay, without further ado, we are running up at the end of our webinar time. Dr. Balko, I want to thank you again for your time today. Victoria, you as well. I really appreciate it. And 3-Day family, thank you guys for taking the time out of your week. We will be following up with an email with some resources for you as well as a survey. So if you would mind, please taking some time and doing that, I'd appreciate it. And also any questions that were submitted that we didn't get to answer today, we will also be emailing out with the responses for all of you to have for reference. So everyone, thank you so much. Please have a happy rest of your week.